So we'll talk a little bit about what a postdoc in the Moody lab would look like here at BYU. I'll start with first the project, and then I'll tell you a little bit about the environment at BYU and some of the application requirements. Uh, Firstly, uh, one of my long-term goals is to make high-resolution protein structure determination faster and easier and more reliable. And so uh, one of my current objectives is to uh, crystallize or to make it possible to crystallize every stably folded domain in UniProt under 50 to 100 kilodaltons. Okay, so kind of in that regime where cryo-EM can't get to the really high resolution. And so I'm currently investigating and have been for the last couple of years something called TELSEM fusion crystallization. Here I'm going to call it 1-tel fusion crystallization. And if you haven't heard of that, I'll just quickly introduce it with some pictures here. So we have the crystallization chaperone is this guy in pink, and it's been genetically fused to a client protein. Okay, at high pH, this thing is soluble, it's monomeric, but as you lower the pH, the 1-tel carrier polymerizes, and it drags that client protein along for the ride. And what happens, and this is the beautiful thing, is that these polymers associate, they zipper up because they're making the same interaction over and over again. Okay, and so 1-tel has proven effective at crystallizing maybe half a dozen genetically attached client proteins, but there is an important caveat here that there has to be an effective linker between the carrier and the client protein. And so just to make that a little easier to see, I put a couple of images here. So we've got the carrier in pink. We've got a helical fusion to this client protein here, this UBA domain. Here we've got a flexible connection. Okay, so this one can bounce around a little bit relative to the carrier. And so the reason that I'm looking at the Rosetta community for a postdoc is that I expect that Rosetta can model and identify those connections that would be most likely to result in a well-diffracting crystal. And so I wanted to highlight a couple of projects. This is not an exhaustive list. I'm very interested in the development of a postdoctoral researcher towards their own long-term goals as well. But potential projects here would be things like sampling different positions along this alpha helix right here to present different faces between the polymers that associate. Another project might be to sample possible docked configurations of this client protein against the polymer and be able to score them and decide which would make the best crystal contacts according to Rosetta's scoring function or some other appropriate metric. There's a possibility to develop novel analogs. For example, TELSEM has 6-fold symmetry along its helical axis. We think there'd be a lot of advantages to having something with a 4-fold symmetry. And then we do some drug and biologic development as well. And so there would be opportunities to develop biologics using this skill set. And so what I'm going to do now is switch gears a little bit and tell you about what it's like to work in the lab and what it's like to be at BYU. So I put a couple of links here. One is to the BYU Honor Code and one is to the application itself. Make that easier for you. I've got my contact information at the top as well. But BYU and I just had to show our mountains, which, by the way, are much more impressive from the ground rather than from a drone. But I thought I'd start with my goals for the postdoctoral position. And for you, if you're fortunate enough to join us, one of them is that you will train and mentor other lab members in using Rosetta and related modeling tools. And as I said before, one of my goals is that you apply the power of Rosetta modeling to the development of this 1-tel crystallization method. But equally important is that you develop as a researcher and that you land your dream job after this postdoc. Okay. And so I thought I'd probably end with some useful facts that I would ask if I was looking into a postdoc position. Okay. I've been at BYU 8.5 years. The lab typically has 2 to 4 graduate students and 11 to 14 undergrads, so we're very undergrad heavy. Oh, and there's a picture of many of us for your viewing pleasure. We have a fairly diverse group, which has been great. Um, you would be my first postdoc, which is exciting. The position is funded by Amira. Uh, BYU puts a maximum of 5 years on the position, so I say that so you can see that the funding is stable, that this could be a little bit longer term. It renews every year. Let's see here. The position is full time. I've pulled this wage from the NIH guidelines. It's salaried. It comes with medical and retirement benefits as well. Okay. And then you should take note of this. So BYU is sponsored by the Church of Jesus Christ of Latter-day Saints, and that comes with a couple of important strings. I should point out, though, you don't have to be a member of the church to work for me or to work here. I've had students who are Hindu and Buddhist and Muslim, many different denominations. But you do have to agree to the BYU Honor Code, which I've put right here. But I'll just briefly tell you that it covers things like dress code and substance use. There's a moral code that's part of it. And then if you go to our application, you will notice that you'll be asked to submit a one-page statement about how you would further BYU's unique mission. Okay, so if you're interested in the position, if you're interested in coming here to BYU, definitely take a look at both of those links. And I'll stop right there.
I think we had some good questions on Monday. And one that I really liked was like in terms of thinking about your lab as like engineer, like using some of the protein design tools for more engineering or kind of like understanding what these models or tools are learning. Kind of where do you see your lab falling on that spectrum?
Yeah, this is a great question. We're definitely an engineering type lab. We engineer biologics. We see protein crystallization as an engineering problem. Um, and for this position, this postdoc position, one of the things that I hope will happen is that a postdoc will engineer software that, if successful, could be commonplace. I could imagine going into UniProt with your favorite disease protein and going down to the structure page, and there's no structures, but there's a little link that says, okay, here is how to make a TELSAM fusion pre-calculated for this domain, and here's a protocol for purifying and crystallizing it.
It's good.
Awesome. All right, so I get a lot of questions about kind of what is the Flatiron, the Simons Foundation and the Flatiron Institute. We are kind of relatively new. We are a non-academic, non-profit research institute. The Simons Foundation was started in 1994 by Jim and Marilyn Simons, primarily as a way to fund external research groups. But about 10 or 11 years now ago, they started an in-house research group called the Flatiron Institute that houses about 200 or so PhD level scientists working in computational mathematics, astrophysics, quantum physics, neuroscience, catalysis, and biology. And so I'm a research scientist and co-head of the biomolecular design team and the Center for Computational Biology. And the Flatiron Institute is located in the Flatiron neighborhood of New York City, right next to the Flatiron building. My group here has a variety of projects that we work on. We tend to kind of work on things other than proteins, so like heteropolymer design, for example, peptide foldmers, where they're your side chain functional group branches off of the backbone nitrogen rather than the alpha carbon. We do a lot of work with cyclic peptides, showing us to kind of a slide here from one of our postdocs, Xiao Zhu, where she developed a new technique to be able to model much larger cyclic peptides than we have been able to in the past. And she's using that to design binders for matrix metallo protein, MMP8, matrix metalloprotease 8. We also really care about the dynamics of proteins and actually trying to engineer specific dynamics and motions into protein structures. So we've been working on designing hinge proteins with a single degree of freedom, um, as well as some kind of flexor proteins, we're calling them, with kind of one potential degree of freedom of motion. But actually kind of trying to engineer the motions, not just finding things that have motion. We try to improve on kind of our current scoring functions and force fields by directly incorporating quantum chemistry calculations into protein design. We have a new package that'll be coming into Rosetta soon called RosettaQM, where this is in collaboration with Ben Brown and Jens, where we've kind of turned Rosetta into sort of like a QMMM force field where you can have score proteins using quantum chemistry, but kind of do it by zones where you could potentially, if you're designing, say, an enzyme, have a zone where you're using extremely high level of QM theory, and maybe the first shell you use a semi-empirical value, and then using kind of Rosetta force field for the rest of the molecule. We'll have a paper coming out on this soon, but when you do start to look at QM models, you can definitely make some improvements to Rosetta. So we can actually predict the aqueous and non-aqueous structures of cyclosporin now, which we couldn't do before with Rosetta. And then we also kind of like to push what we call the tractability ceiling. So this is primarily the work of Victor Mulligan and some other folks in our group, but we're starting to design proteins on quantum computers because the protein design algorithm maps directly to a cost function network optimization problem that runs well on current generation gate-based quantum computers and quantum annealers. Flatiron Research Fellows, that's what we call our postdoc positions at the Flatiron Institute. I think have a fun time. We're, like I said up front, we're not an academic institution. So that means there's no graduate students, it's just postdocs. That also means there's no teaching requirements. There are no grant writing requirements. All of your salary and your research funds are funded by a very large endowment that we have. That does, however, mean that there's no opportunities for translational grants, things like NIH and K99s. And there are potentially fewer opportunities for grad student mentoring. There are definitely potential for mentoring grad students in the labs of our collaborators. And we have an intern program that's very, very popular. We got more than 600 applicants this summer. And FRFs from our group and from the center as well do go on to careers in academia and industry. FRFs, because you're not spending your time teaching and writing grants, we do give you kind of a lot of independence and autonomy, and we really stress collaboration within the center and also kind of between centers. And we actually just started a new inter-center collaboration called COMPASS, which is the collaboration on molecular physics and sampling strategies, which is with basically all the folks at the Flatiron Institute who care about molecules and atom level interactions. And so this is a collaboration with other groups in the Center for Computational Biology, as well as the Center in Computational Mathematics, and kind of our new initiative on computational catalysis. Positions here at the Simons Foundation and the Flatiron Institute are for 2 years, with a possible 1-year extension for good behavior, I like to say. There are housing subsidies for postdocs. There's free lunch, and salaries here start around $95,000. And I should say that we are going to be looking for postdocs to start potentially in June 2026. So I will be sure to, the ad has not gone up yet. It'll probably go up in a little while, but I will be sure to share it on the Rosetta Commons Slack and job mailing lists. Or folks can get in touch with me with my my email address below.
So I have a quick question. How does this is actually more curious about your research, which is that how does RosettaQM do on like active sites and metals and things that Rosetta usually, maybe it's just by design of just how the like maybe fast relax is designed, but just these things usually Rosetta cannot really resolve in any way. And I saw you guys using DFT, so I'm like, oh, maybe this new tool will be able to do that.
100%. That's one of the things that we're planning on using it for, is to try to design heteropolymer catalysts in non-aqueous solvents. That's kind of our 5-year plan. And so the idea is that we've added some new functionality to Rosetta called a multi-score function, which allows you to score different parts of your pose with different energy functions, and does all of the bookkeeping about which parts should be scored with which energy function, and to kind of sum things up the correct way. The RosettaQM creates a bridge between Rosetta and I think it's up to about 3 different quantum chemistry packages now. And so, yeah, you can, just like you can with QMMM, you can create a QM zone where you can define a particular level of theory that you want to have the energy evaluated at. RosettaQM can also do QM-based geometry optimizations, and we map Rosetta constraints onto those as well. So you can also use it for parameterizing things like rotamer libraries or backbone potentials, crosslinks.
Cool. Yeah, thank you. I guess I forgot about the aqueous catalyst thing. I think you mentioned that before, so sorry about that. But it sounds like an interesting project. It's interesting that you can compare like different score functions and stuff. I didn't realize you could do that on a
You
single
can't
program.
you can't yet. It's not checked into the main repo, but it will be soon.
Okay, cool.
Hi, can you hear me?
Yes.
Great. Thank you so much for the talk, Dr. Renfrew. I have been super interested in Flatiron, and I was just curious. I think you've touched on this during your talk, but about in terms of the different groups at CCB,
hmm
would you say that there's like a significant amount of overlap in terms of the amount of collaboration you do together on a given project, or is most of the work being done independently in each group?
Sure. Within CCB, we tend to collaborate mostly with another group called the Structural Molecular Biophysics Group, which is headed up by Sonia Hansen. They tend to focus on molecular dynamics, and also looking at protein heterogeneity from cryo-EM data. And they've done some really, really fun work there. We've collaborated with them quite a bit. They're part of this Compass collaboration that I mentioned. And so a lot of the projects where we're trying to look explicitly at protein dynamics and trying to figure out ways to kind of understand the dynamics or model it, we collaborate with them. Other groups within CCB, we probably collaborate with less so, but we're looking for ways to do that. There's groups that look at kind of large-scale biophysical modeling of things like microtubules, some developmental dynamics, biological transport, and there's and a handful of other groups. So, and also the large genomics group as well.
Great. That's great to hear. Thank you so much.
I had a question, if that's okay.
Sure.
So what type of background are you guys looking for? Like specific computational, like PhD, or just like a biochemistry PhD?
So yeah, and when we're looking for postdocs, we're looking for folks that have, I guess we do a good bit of software development in our group. One of the kind of the missions of this Flatiron Institute is to simply kind of do basic science, but also to kind of be a bit of a repository for knowledge, but also for software projects. Oftentimes, in academia, not so much with Rosetta, but a grad student will start a project, and then that software will work for a few years until the graduate student leaves, and then the documentation becomes out of date, and the project just kind of festers, and someone has to rewrite it a few years later. We try not to let that happen at the Flatiron Institute, and so we actually have a whole scientific computing core that also includes a lot of specialists on software development, as well as kind of maintaining an internal computational cluster. So we do tend to look for folks that have a bit of a software development background. That being said, we've often found that it's easier to teach people coding than it is to teach them the physical chemistry and the biochemistry.
Okay.
Particularly when we have such a kind of a large support network here at the Flatiron Institute.
Yeah, so you will say that there's like, because I come from the background of I look into dynamic enzymes that are dynamic, but in cryo-EM. So
Mhm.
like someone comes with that type of background, is there a possibility of training in, those specific areas like computational
100%. Yeah.
dynamics?
Mhm.
Oh, okay.
Absolutely. Yeah. So folks that kind of understand protein dynamics and how different salvation environments affect proteins would definitely be of interest to us.
Okay. Thank you.
Awesome. Awesome. So I'm Parisa. I'm at the University of Oregon, and the project in my lab is wide. It's not like that similar to Doug, actually. So we have projects on peptide modeling and synthesis and design. We do projects like inhibiting proteins, designing macromolecules, designing peptides or proteins to for sensing and delivery, and then also designing new enzymes. But for for this period, I have probably one project of designing the NTP inhibitors that is supposed to bind to them, and I will be seeing the fund soon. So when I saw this, I will post it in a Slack channel and also in Rosetta Commons. I also am happy to take any people that like peptides or designing new enzymes and metal enzymes, but I would say that for those people, it would be easier to write a fund first, and then if you get it, I'll take you. And I think it's like the good things about academia and the bad things about academia, right? If you like writing grants or you want to have the experience, it's good, and I help you. If you don't like writing grants, then it's gonna be more difficult. Then I would maybe think about somewhere else that doesn't have that grant writing. The postdoc in my lab, of course, will be mentoring graduate and undergraduate students. They have one-on-one meetings. They help with grant writings or writing the papers. And then they also attending conferences based on what they want. So it can be conferences that lean more toward academia or conferences that lean more toward industry, depending on what they want. And then you can see this is my group. It's very big right now in this picture, but it's getting a little smaller. And then I think I'm gonna stop and actually let people ask a lot of questions that they have, because as I said, some of the things that I explained are really based on what you want. Yeah.
I do have a question. Do you have a cryo core or some sort of structurally some sort of like collaboration with like if there's opportunity to keep doing some sort of like structural either crystallography or cryo?
Yeah. We do have an amazing crystallographer already that recently joined us, which we are really happy because then we can collaborate with her. So that is a possibility for sure. For cryo-EM, we mostly send our samples to other people that do cryo-EM, but for crystallography, she's right here next to us. So it's a lot of fun, actually.
Nice, thank you.
Do you find that you tend to have more people join your lab from a background in biology or from like computation or like machine learning?
most of the people that join my lab are good at experimental work, so they know the experimental side, but they are lacking the level of computational that my lab uses, because I think, as Doug also mentioned, teaching them the computational aspect is much easier than teaching them the whole experimental thing. So I feel like I'm more on that side. So students come to me, they're very good at doing experiment X, and then I'll teach them how to get to the level of computational thing that they want. Usually that's the case, but I have cases that they were perfect or really good at computational, and then they stayed being computational. So I had both.
Yeah, so it's like open to any questions that anyone can have, anything that would be helpful as you're thinking about postdocs.
I mean, at least for me, I came to talk about my lab, of course, but also in general, I have, if you want, I can give you opinions on what's like postdoc, what should you care about, which lab should you apply, and things like that.
I have a question, maybe not about that, although that sounds interesting, Parisa. Which is I come from a computer science, like somewhat of a physics undergrad. Now I'm like ML for proteins type stuff. And
Yeah.
I use Rosetta as a tool and FastRelac as a tool and like also Rosetta Ligand. But I've always thought about potentially moving into like quantum mechanics work or DFT or uh, MD work, but I've always kind of like thought maybe not because I don't know, I don't have the background like the chemist or the biochemist or the biophysicist or like they might have all that knowledge and I'm sure I'm curious like in the field like are people usually hired? I'm sure anyone could always learn something, right? But like in the field in practice, are people getting hired for those positions usually people who have that computational like pure computational chemistry position or like can someone come in with a or if people have been hired with like that ML background before and they're like, well, we'll teach you all these like niche DFT type work, DFT
Yeah.
type information. I don't know if anyone has a thought
Yeah,
on that.
I can speak a little bit on that, and then maybe Doug and the other people can help a little bit. So, I feel like this is something that's in mind of a lot of people, and this is something that actually causes a lot of them to not apply. But from my understanding and from what I've seen, a lot of people have the potential to learn new things and be excellent in them. And I have a student that studied computer science, for example, or studied something unrelated to science at all, and then went on and learned like biology and computational biology. So I think it's always possible. Just like people talk about it less, and you feel like it's impossible, but I'm telling you that it is possible because I'm actually training two of them. So
Sure, I'll chime in. When we're looking for potential postdoc hires, our postdocs, like I said in my slides, are a little bit shorter. It's kind of it's about 3 years is typically kind of the max for our group, and so we do, and that can be kind of a tight timeline if you're trying to kind of implement some new feature, test that feature, use that feature to maybe design some new proteins, and then try to get that experimentally characterized. That's a very tight timeline. We're trying to get it longer because that's the timeline for kind of all of the different centers. And so it works for the mathematicians, but it doesn't always work for the biologists. But that being said, we do tend to look for postdoc candidates that have particular sets of skills that are in line with a lot of the projects that we already have going on. But we can tailor projects to the skills that you have, with anticipation that you will pick up something while you're here as well. So, if you were coming in with kind of a background in computational chemistry and doing DFT calculations, you might be more aligned to working on some of the enzyme design projects. If you were coming in with a background more on protein dynamics, you might be more aligned to working on some of the projects on designing intrinsically disordered proteins, or the hinge proteins, or kind of flexors.
Yeah, that makes sense. Yeah, thank you both for the answers. So that helps.
May I ask a question to the room? So much like Bryce, I'm also coming from a deep learning background, mostly making deep learning models for protein design, prediction, interactions. And as I've been kind of exploring these postdoctoral opportunities, I'm never quite sure which particular system I want to focus on. I think my skills are fairly transferable to a lot of different things, but I'm never sure if I should just focus on labs doing, say, genomics work if I were interested in that, or protein design work if I were interested in that. And I've just kind of been applying around to different groups doing all sorts of things, and I'm not really sure if that's the correct approach to have when seeking a postdoc. I was curious if you guys had any insight as to whether you should stay focused in your search for postdoctoral opportunities, or if you should be willing to branch out.
I can go on first again. So I would say, in contrast, at least in my opinion, in contrast to the graduate studies, that it's not that important what you study. You just study them and you show that you are competent. What you do in postdoc depends. So if you want to go to industry, maybe a postdoc is not that important as long as you know a general sense of the field. But even then, I feel like you're usually closer to things that you worked on your postdoc in. So I would highly invite you to decide what field you're liking more before you apply, especially if you want to stay in academia. If you don't want to stay, it's a little less important, but I think it's still important, right? My opinion.
Yeah, maybe I'll say just a little bit here. When I did a postdoc, this is a N of one, but when I did a postdoc, I wanted to learn new skills, and so I did something that was interesting to me, and the skills that I brought from my doctorate were useful in that thing, but I also learned a number of new skills that were quite different. And so then I got to have all of those skill sets together as I moved into a more senior position. So it really depends. I think really I would say just think about where you hope to end up, and think about, okay, in a postdoc, most postdocs want you to bring a skill set so that you can benefit the group in some way. But at the same time, you have a right to expect that you're going to learn something new. I would say if you go to a postdoc and you don't learn anything new, that wasn't very useful, right? And so, yeah, I think it's a little bit of give and take either way. But definitely, if you just have a sense of where you want to go and what things you want to add to your skill set, your repertoire, that'll help you kind of narrow down where you're looking.
Great. Thank you guys for your input. I really appreciate it.
I wanted to ask one more question. I just wanted to know what specifically makes a postdoc application stand out. Like, is a particular set of skills, or the success of their project? Yeah, just what really helps you to stand out.
I'll take this one. We just went through a hiring phase for a joint postdoc position. And so typically the things that I look for are someone that's done a little bit of homework on the position. For this last position, we got about 60 applications. And about 25% of them were just from people who had applied everywhere, and were kind of very generic. The ones that we tend to focus on the most were the ones that they could have mentioned the PIs in the cover letter and kind of have particular projects in mind, or maybe describe how their graduate work could apply to some of the work that is happening in the group. So folks that have kind of put a little bit of thought into what it would be like to potentially work in that group.
Thank you.
Yeah, I would say it's exactly the same as Doug mentioned. We all look at like all the things he said, and also for myself, I also look at the students and how he showed already that he wants to be involved in teaching a mentor, because most of the postdocs in my lab will have a mentor. So I want the postdoc to be interested at least in jumping to teach a mentor. And when you have all of this, then you become a member to then ask you some questions over Zoom. And then if you pass that one, then we invite you to come have a look. Yeah.
Yeah, our process is similar. I think we could have narrowed down our list to about 10 candidates or so that we did short Zoom interviews with, and then we're going to be bringing in probably about 4 or 5 for in-person interviews.
Definitely, if people have more questions, feel free to jump in. But also, I'm wondering if the three of you had to give just like general postdoc advice, or maybe things that you considered after graduate school that you would kind of advise other people to think about, kind of what would be your top tips?
I guess I can go first here, and this will be similar to what Parisa and Doug have said. You want to show your potential employer what you can do for them. Like, what skill do you bring? And I agree, you definitely want to show that you've looked into what they do, that you're passionate about their projects. What ideas do you have to push their projects forward? I think that will make you a strong applicant.
I can go next. I'm gonna tell something to you that is now it looks funny, but at the time when I was toward the end of my PhD, I started feeling that I don't like this project at all, and I want to completely shift what I'm doing. And I applied to a bunch of high neuroscience labs, and none of them replied to me. And then I was like really sad and went and talked to my advisor, and he said, "Why don't you try to apply to 5 people that's in our field?" And then I tried to apply to 5 of them, and all of them replied back to me. And I was like, oh, okay. So maybe I have to stay in this line. And now I'm really happy. I think at that time when I was at the end of my career, I was just so tired. So tired from applying, and I didn't want to do anything with any of this. So I didn't but when I had 3 months of relaxing and then going to the postdoc, I found out that I really like this. And I was also really happy that I chose somewhere that was like computational biology, so that I also could use my computational skills that wasn't very usable back in my PhD. So I would say that sometimes you may feel like I'm going to 100% change the field and go somewhere that blah blah blah. And sometimes think about it twice and give people that are closer to your labs also some thinking, because they might be much better than what you think, and you're just not in their mood back then.
I think my advice probably would be to kind of really think about the type of person you are and how you work, and kind of what you kind of really need from like kind of a mentor and advisor. Like when I did my postdoc, I really liked the lab I did it in, because I had kind of a good bit of autonomy and kind of a relatively long leash, so to speak. I would not want to have been a grad student in that lab though. But I felt like it was a really good fit for a postdoc for me. primarily because of the I knew I wanted a little more handholding and a little more direction as a grad student than I did as a postdoc. And so trying to find kind of a postdoctoral PI or mentor that will kind of allow for that and not micromanage you and not try to shoehorn you into a particular project, I think would probably be key.
I'm gonna just tell that 100%, Doug. It's really important that the people you do your PhD with and the people that you do your postdoc with, they're very different. And you need to look at the postdoc as something like something more open, something that allows you to practice your next period of life. So, yeah.
Yeah, that's an interesting point from both of you about the post. I also have a professor right now who I definitely like the hand holding and guidance, but definitely thinking about that switch as I move on to the next career option. That's important because that's like very different than my current professor.
Yeah, my postdoctoral PI was, I wouldn't describe them as hands off, but they ran their lab in a different way than my graduate PI. And so yeah, just finding a style that meets you where you at or where you want to be, I think is key. So, and one thing would be, to most labs have websites. It's easy to kind of reach out to other folks who are already in that lab and get a feel for what it's like to work there. If it's another lab within the Rosetta Commons community, there might be on this in the Rosetta Commons Slack. You can probably find them on LinkedIn. It's relatively easy to get a feel for what a lab is going to be like.
Yeah, the Rosetta Commons is pretty helpful too. I can't believe the connections I've made just through Slack. It's crazy.
That's right. I also wanted to tell that apply for more than one lab. If you have more options, you have more chances of choosing, and I think that's important.
I think one thing that came up in the last session as well, which maybe if folks are already all part of the Rosetta community, you're maybe more aware of it, but like maybe for the faculty, if like you could highlight maybe unique things that just being part of the Rosetta Commons, like being in a lab that is part of the Commons, like kind of unique advantages it might have for training or for postdocs. Because yeah, I mean, it is kind of a very unique community.
Yeah, I think it's definitely unique. It was, I know when I was a grad student, it was particularly advantageous at the time. Like I say, I'm a Rosetta lifer. I worked in Brian Kuhlman's lab as a grad student and then Rich Bonneau's lab as a postdoc. Both of them were in the Rosetta Commons. But Brian only had, when I joined Brian's lab, we were transitioning from Rosetta from Fortran to C++. At the time, Brian only knew the Fortran code and didn't know how to program in C++. I did not know Fortran. I did know C++. And so, Brian could tell me where something was in the Fortran code, but not how to do anything in the C++ code. But I had the Rosetta mailing list. And so I had this kind of whole super lab that I could access. In addition to Brian was also a new PI. So I think there was only 2 other grad students in the lab when I was first starting in Brian's lab. And so, yeah, having access to this huge community just when you don't know how to do something was incredibly useful in my graduate career. And postdocs, and now it's fantastic for networking. RosettaCon, and the internship programs, the boot camps, all of them are kind of provide a tremendous amount of value for the community.
Yeah, I mean, I agree. I came from a lab that wasn't part of a Rosetta community lab, and then I came to David's lab, and it was became a Rosetta Common, and I mean, there are so many help and advice and so many ways to find answers to all the things. It's unbelievable, and I'm just really happy to be part of this. And I'm like And it seems like it's a community that keeps people together, because whoever whoever is studying there stays there. And I think everyone is happy about that. So I have for sure happy to be here. And as Doug said, also they have other resources for travel, and then small mini grants, and all the things that you want, that in addition to the mechanisms that are in that you can find, they have also their own pot of money that they can give you. So that's also really good.
Okay. Unless anyone has any final questions, I want to thank Doug, Parisa, and James for being here and sharing about their labs and some of their thoughts on postdoc positions. Yeah, I think everyone had their emails and their slides, so feel free to reach out to people. But thanks everyone for being here. I thought this was really fun